Sure. Ese es de la Interamericana. Amarillo y verde. Así que vamos a sacarlo de aquí. Buenas tardes a todos. Para mí es un placer ser moderador de este seminario. Tenemos hoy al doctor Brad Wizard. Brad, he comes from France and also California. He moves back and forth during his childhood with his parents. So he's fluent, not just in English, but also in French. Basically, French is his first language. And if you want to talk with him, he can actually understand you in Spanish. So if you have questions, and you need to speak slowly. Brad got his bachelor's in science from University of California, Los Angeles, in 1993, and then his PhD in 1999 from the same university. He just published 10 articles as a graduate student, just 10. And he was awarded 2 fellowships during his graduate studies. After that, he moved to UC San Francisco, where he was a postdoc from 1999 to 2002, where he has published there 9 more articles. So he's a very prolific young scientist, and I'm very happy that he's in Puerto Rico and that we are going to start a collaboration here at the University of Puerto Rico with UCSF. So here's Brad.
Can everyone hear me okay? How about now? Yeah, it's on? Great. So I wanted to thank you all for giving me the opportunity, first of all, to come and share with you something that I'm really passionate about. I love human genetics, and as you'll see, I found a way to involve myself in a lot of different human genetic disorders. So even though the focus today will be about our new proposal in collaboration with UPR, which is involved with genetic determinants of lipodystrophy in HIV-positive patients, I wanted to kind of give a perspective of how this study fits into my program of research and into human genetics in general. Once we talked about the pilot, I wanted to then focus in on some new methodologies that are going to help investigators, such as myself, that are looking at more and more gene variants, right? We'll be looking not only at a few, but getting into the hundreds, if not thousands, of genetic variations. So some methodology that we can use both at the bench to collect the data, so some high throughput genotyping methods, as well as some data analysis methods, because even though it may not seem like a big deal, you can actually spend a whole lot of time analyzing even one polymorphism. And so when you start looking at hundreds of them, that really consumes even more time than collecting the data itself. And then I'll entertain questions. So one slide that always puts things into perspective for me is this one right here. I think one of the most important paradigm shifts that we've seen in our generation is the fact that human biology is no longer an issue of nature versus nurture, as a lot of us were taught in school, but that really it's a question of nature and nurture in combination. What that really means for scientists is that all diseases have both a genetic and an environmental component influencing expression. And the issue is, or the question really is, how much is genetic and how much is environmental? And that's really well described by this image here, where you have really 3 classic examples that demonstrate the spectrum. On one end of the spectrum, we have diseases that are almost entirely programmed by our genetic code. Those tend to occur very early in life. They tend to be pediatric diseases. One example is cystic fibrosis, where there's very little that can be done in terms of manipulating our environment to improve outcomes for these affected children. On the other side of the spectrum, and really the focus of today's talk, are diseases that are almost purely environmental in nature. And those tend to be pathogens. That's almost the easiest one to think about because that's literally something from outside that infects you and impacts you. However, we have already seen in the literature that there's some amount of innate genetic factors that allow us to really help minimize or sometimes accelerate the course of a disease. And that's really what we're interested in using. And the majority of diseases that I work on in my group deal with those diseases that have pretty much a fair mixture of both genetic and environmental components. These are problematic because they're very complex and unfortunately they're very common. That's generally speaking what most of us in this room will succumb to in our older years. So we're talking about adult onset diabetes, we're talking about cardiovascular disease, arthritis, cancer. Anything that's very common tends to have many genes, many environmental determinants, and they all interact to increase our risk of disease, or in some cases, protect us. So really the goal of the Human Genome Project is to leverage our knowledge of genetic programming to benefit patient care. But one really important cautionary note I might add, particularly because there's so many students in the room, is that a lot of people think the Human Genome Project implies that we're only interested in our genetics and that genetics is the solution to everything. And that's simply not true. The reality is we want to understand as much as possible about our genetics so that we can then go back and understand more about the environment and then use both pieces of information to maximize the health of the patient. Right? So the old paradigm of genetics, really what you see in the literature now, if you go ahead and look at human genetic disease, has been based on the concept of monogenic disease. So that's, you have diseases that are caused by single gene mutations that are they're very powerful. They're what we call fully penetrant. And the important note here is that although those were important to help define the science and kind of understand the mechanism of disease, when you group them all together, they account for only about 10% of the diseases that we see in the clinical setting. And more importantly, they're not that common individually. The other 90% are what we call these complex and common diseases. So just what we were talking about, diabetes, heart disease, the whole bit. Now, the important understanding here is, even though at the beginning people were very pessimistic about being able to identify determinants for these diseases, now we're learning how to pick apart the puzzle one piece at a time, and we've come to realize that there's a central concept that we can exploit, and that is individual genetic determinants may have a small impact, but that's okay. Now that we understand how to design our experiments, we can actually find small genetic changes that increase your risk of disease, go back and then slightly change the design of the study to find the next incremental determinant, and so on, and keep going, and then eventually start modeling how all of this comes together to make a much more powerful impact on your risk of disease. So the goals of this pilot in particular are to hopefully improve outcomes for individuals being treated for HIV and AIDS. So we're not really looking at how individuals become infected and if they might be resistant or susceptible. We're more interested in identifying what genetic determinants might help to predict some of the outcomes based on the very aggressive antiretroviral therapy that causes a lot of secondary sequelae. Of course, a goal is to decrease health disparities in the affected populations, particularly given that the spread of HIV is just booming in all populations right now. And health disparities is a particular issue when you have a disease that requires a lot of medical attention, and that's very expensive to do. And of course, our interest in forming collaborative relationships between investigators both at UPR and at UCSF to trade technological as well as intellectual resources, which is something that's always a lot of fun for an investigator. So I wanted to give you a little bit of background about how I found myself working on lipodystrophy, because that's definitely not my background. I'm a geneticist first, and I found myself involved in heart disease research early on in my doctoral work. I was working primarily with familial combined hyperlipidemia, a complex form of hyperlipidemia which is pretty frequent. It happens in about 1% of the population. 1% doesn't seem like a big number, but when you consider how many diseases are out there, it's actually the most common form of dyslipidemia, and it leads to a 10-fold increased risk of premature myocardial infarction. So you will get a heart attack, and on top of it, you'll get it much earlier than the average age of about 65. So it's a very serious disease. As a graduate student, I started a series of candidate gene studies based on what we knew about lipid metabolism and tried to identify variations that might be enriched or depleted in individuals with the disease versus controls. And I was doing this both in a case control design as well as in family studies. That went very well. It was very productive, and I'd found many genes that were enriched in this disease. And then as a postdoc, I was working in a really interesting, highly collaborative clinical setting at UCSF, interacting with a lot of clinicians, where I had the opportunity to start working with a clinician scientist that was interested in fatty liver disease. So this is a non-alcoholic type of fatty liver disease that ends up very often in cirrhosis, and then that requires liver transplant. And they approached me to actually mentor them about doing genetic studies. In the course of the literature review, I started to realize that the rationale for a lot of the same candidate genes was actually present for liver disease as it was in heart disease. And it seemed kind of confusing at first how something that was impacting two very different organ systems could be related. And that rationale actually became clearer a little bit later. So I tested the same genes in this new population and realized that there was indeed an overlap. Two genes in particular, peroxisome proliferator activated receptor alpha, or PPAR alpha, and microsomal triglyceride transfer protein were both enriched in both populations. And the other two genes, lipoprotein lipase and carnitine palmitoyl transferase 1, these are all genes that are involved in fatty acid metabolism. Now, of the genes that were shared, the first point of interest that I found was that clearly we might want to study those genes that are involved in several diseases, because that means discoveries that we make about how to maybe even come up with drugs or change therapy based on that genetic information could help potentially a much greater pool of individuals. And even within that group, I realized that there's another advantage to prioritize, and that is some of these genes, because science doesn't move in one direction, it moves in every direction. One of these genes in particular, PPAR alpha, which is a transcription factor, already had a pharmacologic agent that was used in the clinical setting to control hypertriglyceridemia and improve HDL cholesterol. So here you have a gene that already has a drug in the market that directly interacts with that gene, okay? And now you have the opportunity to identify variations which could tell you how well a patient might respond to that drug, or perhaps maybe they won't respond at all. So already you have something that's already ready to translate from the bench side to the bedside. So I realized that I should not only look at determinants that were specific to disease, but prioritize those that were common to many diseases, and within that, look for genes that were already close to some kind of translational research. So from there, I started courting interactions with a lot of other clinicians that had different patient populations based on my literature review that looked like they could share some genetic determinants with the disorders I was interested in. And long story short, there were 5 diseases I found that I could actually leverage and work with. So that includes looking at primary obesity, primary adult onset diabetes, and finally lipodystrophy. And if you think about how much time and money it takes to recruit patient populations and maintain them, it's very attractive to investigators to be able to do something with this population outside of what their primary program of research is. So you can really, you can get a lot more mileage for the same amount of money. It's very hard to maintain these populations and to be funded by the government to collect them. So I think uniformly, every investigator I've talked to has been very attracted by the idea of using their populations in an additional way, as well as the actual subjects are very happy to contribute in a more substantial way to scientific discovery. So about this time, my departmental chair, who was trying to corral me and make sure that I wasn't going in every direction at once, said that, and rightly so, she was very sharp in that respect, said, look, you have to commit to some kind of centralizing idea, and I didn't really want to give everything up, and it ended up being a very productive exercise. In essence, I came up with the following diagram, and that is that in terms of genetic determinants of disease, you can really think about determinants in several kind of classifications, right? you can have specific determinants at the top. These are genetic changes which predispose you to specific disorders or metabolic defects. And I listed a few of them for you. And then underlying that, you can kind of cluster genes into those that may involve, for the sake of lipid metabolism or energy metabolism, genes that influence uptake, genes that influence storage, genes that influence disposal. And you might call those, let's say, thematic determinants that can then predispose you to a couple of these disorders. And then underlying that, you may have a very small subset of genes which more generally impact many facets of energy metabolism. And those will tend to be, for instance, transcription factors, right? PPAR alpha, as luck has it, is just one of those general determinants. It only has a few other family members, and together they really have a powerful influence on lipid metabolism throughout the body. So in that sense, I was very lucky to have that kind of data. And really what my lab is concerned with is identifying all of those. So we test candidate genes, and then we fit them into this scheme of how general or specific they are to help prioritize subsequent research projects. And so by satisfying my chair, I really was able to come up with a really nice theme as to how I should conduct my program of research. And that leads us into the new sector, which is lipodystrophy. So a lot of you might ask, why then is innate genetics important in HIV, particularly given the first slide where I showed you that really HIV is primarily an environmental determinant of disease, and that there's very little that we know of that can impact susceptibility to HIV. And again, I remind you that we're not interested in looking at those individuals that are resistant. That actually is a productive line of research, and there's drugs being tested based on that information. I'm more interested in the fact that most individuals don't have these resistance traits. And so they are susceptible to HIV. And once they are, what kind of genetic determinants will help predict how they'll fare on these regimens that really lead to serious metabolic conditions and also physically disfiguring, which makes it very difficult for these patients to stay on their drug regimes. So even though we've seen favorable virologic and immunologic and the clinical impact of highly active antiretroviral therapy, or HAART, these adverse drug reactions, peripheral lipodystrophy, central adiposity, dyslipidemia, and insulin resistance, occur in up to 80% of individuals on these drugs. So that means the majority of these people that are now living a long time again are bound to get some combination, at least one, if not all, of these complications. So again, they're going to suffer from secondary effects much earlier than anybody else that doesn't have to deal with this pharmacologic burden. Okay? And interestingly, pretty much all of these secondary or these comorbidities are attributable to components of these therapies. There's a small subpopulation that looks like they may even get a subset of these comorbidities without drugs, but that literature is a little bit murky. The last piece of information which suggests that innate genetics might be important and possible in HIV is the area of pharmacogenetics, right? Now, there's enough in the literature that shows us that you can look at genes that not only impact how you might metabolize a drug, and so those are really primary genetic determinants of drug metabolism, but there's a series of other genes which might influence how well a body could tolerate these metabolic changes that are going on. If you're on a drug that damages your liver and changes your lipid metabolism, there are certain genes that allow you to tolerate that kind of hyperlipidemia much better than other variations. And that's really what we're hunting after. So there are several phenotypic clues to suggest candidate genes that might be interesting to look for variations that are common that might be useful in predicting who might suffer from these different comorbidities. Clearly in hyperlipidemia, which we know a whole lot about, the fact that these individuals develop increased LDL cholesterol levels and increased VLDL cholesterol levels, so these are lipoproteins that are risk factors for heart disease, and increased levels of apolipoproteins B and E, these are actually proteins carried on the lipoprotein particles, and they're actually very powerful markers of dyslipidemia, or they can be. And the fact that individuals develop dyslipidemia, which independently of the HIV field, there's been progress made in inherited forms of lipodystrophies, which are rare, as well as benign symmetric lipomatosis, that suggested there might be a pathophysiological overlap, and that those genes in the inherited forms, instead of looking for the knockout mutations, you could look for more modest and common variations that in those individuals that aren't on any kind of therapy tolerate. There's not really a very easily distinguished metabolic difference. But when you stress somebody with these drugs that really hurt the liver and change lipid metabolism, might be more fully expressed. And of course, there's the observation that cytokines and hormones impair fat and glucose homeostasis in patients that are receiving HAART. So our proposal focuses on the following. So given that lipodystrophy is a risk factor for cardiovascular disease, steatohepatitis, so fatty liver disease, which leads to cirrhosis, and various insulin-resistant states, metabolic syndrome, type 2 diabetes, all of which decreases adherence to treatment regimens and increases the cost of management. And a further burden is probably the fact that many individuals seek elective cosmetic procedures because lipodystrophy can be disfiguring. That we aimed this pilot to explore the impact of genetic variations in drug metabolism and fatty acid metabolism themed genes in HIV-positive subjects and their effect on outcome variables related to intensity of drug regimen, frequency of regimen modification, impact on frequency, and measures of lipodystrophy, which can be used then to justify large-scale searches for genetic modifiers of HIV treatment and outcomes. So the central hypothesis of this proposal was that this study is that there exists a limited set of genes that modulate HIV treatment by impacting the effectiveness and time course of treatment regimen and risk of secondary sequelae. There are three specific aims in this proposal. The first is simply to screen 250 HIV-positive subjects with in-depth patient records for variations in 8 genes. The second is to compare polymorphism prevalence when the population is dichotomized with respect to lipodystrophy pattern, drug regimen, or quality of life measures. And then the last aim is then to assess the functional significance of the polymorphisms through correlation with physiologic measures that we collect. And thereby we attempt to elucidate the mechanisms by which these variations may affect disease expression. So there are 8 genes that we propose in this first pilot study. And if you review the literature, there's actually many more that were suggestive. And in order to prioritize those genes as those most likely to be yielding productive data so that we can go ahead and go for a much bigger grant, we decided to focus on those genes where there was multiple lines of evidence. So in the case of these 8, 2 of these genes in green actually interact with proteins that are expressed by the HIV virus. So there's literally a direct physical interaction. And all of these genes have been studied at least biochemically, so they either impact the mRNA or the protein expression of these genes. And a few of them, since we submitted this pilot, actually there's some genotypic data, and these polymorphisms that are on the far column, a few of them have actually been studied in much smaller pilot studies. So there's some suggestive data that they play a role. When I selected these polymorphisms, I made sure to prioritize with three criteria. The most important is to look for very common variations. And the reason being, when you have a small pilot study, you need to use a common variation that you have a chance of actually seeing that variation in your population. Okay, and so that gives you a little bit more power to do your statistical studies. And then, of course, if they had been studied in any pilot studies in this similar kind of population, and, of course, something that prioritized polymorphisms is if there were previous studies in totally different populations that show these polymorphisms were functional. So if they change the amino acid sequence, like PPAR gamma, so there's an amino acid change at position 12 from proline to alanine. So clearly that's a much more attractive candidate because we already know that it actually changes some of the properties of PPAR gamma's function. Okay? And those were the 3 criteria we used. So in terms of the study populations, I thought it was worth kind of describing it. At UCSF, we'll collect 150 subjects. We've already started enrollment. We have 15 in hand. The demographic breakdown of the clinic, it's actually the liver center at UCSF, tends to be approximately 85% non-Hispanic Caucasian, 9% non-Hispanic African descent, and 4% Asian. And then at UPR, we'll recruit another 100 subjects of Hispanic descent. The variables we'll collect in this pilot study are lipid profiles, so looking at total, very low density, and high density lipoprotein, cholesterol, and triglycerides, free fatty acid levels, glucose and insulin, and of course a lot of the other measures that are recorded in patient charts: height, weight, temperature, pulse, blood pressure, wasting and fat redistribution, so the lipodystrophy measures, as well as co-infection of hepatitis and any comorbidities that are recorded. We'll also take into account opportunistic complications, not so much for this pilot study, but in anticipation of recruiting a larger pool of patients where we might be able to look at these variables as well. So now we get to the methods, the high throughput. So using these three aims, these are the methods that we'll employ. I'll start with aim 3 because that's the easiest to go through. For the data analysis, we're using pretty standard techniques to look at associations between a categorical variable, right, and a continuous variable. So we'll use a ANOVA and t-test, as well as non-parametric tests. But for aims 1 and 2, that's where we're introducing some new methodology. For collecting the genotypic data, we're using a new technology developed at UCSF, actually, fluorescence polarization single nucleotide polymorphism detection. So FP-SNP is a lot easier to say. And this works about 95% of the time. So it's a pretty robust assay. And for those few that may not work, we can rely on the standard technique, which we've all been doing for years and years, which is restriction fragment length polymorphism assay. And that'll definitely take care of the other 5%. So that's one methodology I want to talk about. And then some new methodology in terms of analyzing genetic data is the other method I'd like to talk about. So, of course, primary genetic associations, so looking for how a gene may differ in cases versus controls, is pretty straightforward. We tend to use a chi-square test, or if the sample size is small, we use Fisher's exact test. But really, what we want to start looking at is not only the primary genetic effect, but we want to see how these interact. And that can be different. That can be a little difficult to model. And there's a new methodology called set association that I wanted to describe to you. And it's based on the chi-square test, but it uses a little bit more information. So let's talk about FP-SNP. So the beauty about FP-SNP is that it starts with the same basic technique that most genotyping assays start with, and that's PCR. So you can use any, I think most of us know PCR, you use primers, right, to bracket a polymorphism, so you can amplify that area, and so you have enough template to cut it with an enzyme to tell the difference between individuals that have one allele or the other allele, right, and that boils down to a single nucleotide change. So in FP-SNP, you do the same thing. It doesn't cost or take any other different method. You amplify that sequence, and now all you do is carry out a secondary PCR. So before you do that, you degrade the excess primer, and you degrade the excess dNTPs that you add, right, the building blocks for PCR. You add a tertiary primer that sits down immediately adjacent to the polymorphism of interest. And this is where the difference in FP-SNP happens. You actually add the dideoxynucleotides, or the deoxynucleotides that you add, the two alleles that you have, you actually add those two nucleotides, and they're fluorescently tagged with two different fluorescent tags. So they fluoresce in different color spectrum. Okay. You go ahead and carry out amplification, and instead of resolving these products on a gel, which takes a lot more time and money, and probably the most expensive resource is technician time, right? You can actually measure this in a fluid state without running it on a gel, and it takes about 5 minutes to read an entire PCR plate, so 96 samples. Or you can even upscale to 384 samples, or basically as good as your plate reader is and as dense as your PCR plate can be, that's how many genotypes you can collect. So in essence, this is the technology behind fluorescence polarization SNP detection. If you have a primer, right, this tertiary primer that extends by even one nucleotide, it's spinning around in solution, right, spinning around in all directions. So I don't know if any of you have ever sat on a chair that churns, And when you were a kid, you got spun around. And if you pulled your arms in, you spun really, really fast. And if you threw your arms out, you slowed down. Same thing happens to these primers. Okay, so if they're extended, and remember that the fluorescent dye is very large. So even though you only extend by one nucleotide, it's actually much bulkier. So it spins very slowly in solution compared to the single nucleotide that's spinning very fast, right? So if you expose these to polarized light, the molecules that spin very fast will then depolarize the light in every direction, right? Those that spin very slow will tend to not depolarize the light as much, so it will stay polarized. So you simply measure the polarized light in the two colors, and that's how you genotype something in solution. Right? It's a very elegant, very amazing technique. It works very well. And this is basically an example of the output. Now, I have to apologize here. There's actually two SNPs that I was looking at, but this was the clearest digital image that I could cook up. So in essence, you can choose either the pink or the green. It doesn't matter. But you have on the two axes the two different fluorescent dyes that are being read. So if you only see signal in one axis, right here, essentially that means that both copies of the polymorphism, right, because everybody has two chromosome copies, so you have two potential alleles. If they're the same, you'll only get fluorescence in one axis, right? And again, if you have the other polymorphism, if you're homozygous for the other allele, same thing's going to happen. You'll only fluoresce in one color. Now, if you have one copy of each, you'll fluoresce one color from one allele and another color from the other. And so these are the individuals that are in the middle, right? Again, it's a very quick assay. You do a primary PCR, takes about 2 hours. You do a cleanup step, about half an hour. You do a secondary PCR, which is only 5 cycles, so let's say half an hour. So now you're at 3 hours. You put it on a plate reader, it takes about 5 minutes. So you can literally screen anywhere from 96 to 1,500 genotypes in 3 hours. And that's without adding any layers of technology to automate it further, which costs more money. Now, the advantages are pretty clear. It's gel-free, right? That reduces cost. The reagents are universal, so you still do your standard PCR. You can actually pull out old PCR tests that you use and optimize it to the strategy. You don't have to order any specialized primers or specialized chemicals. All you do is order these DNTPs that have the fluorescent tag on it. The assay cost is then very cheap. And again, the most important reduction in cost comes from the fact that it takes a technician very little time to collect an immense amount of data. And that's what makes it high throughput. Is that pretty clear? Yeah? So just to let you know, and we don't have time to get into the other methodologies, I want to briefly say that there's 2 other even higher throughput methodologies that we're exploring in the lab. One is called fluorescence quenching. It's very similar technology where you can actually pool your 2 groups, let's say your cases and controls, into one tube, and you quantitate how much they have of allele 1 and allele 2. So basically you reduce your sample size down to 3 wells, group 1, group 2, and a control. So it's an immense amplification of what you can do in the lab. The idea there is if you screen a SNP and there's a difference in the frequency, you can then go back and individually genotype everybody. But if there isn't a difference, then you don't waste your time with that SNP and you move on. So you'll only spend more money on those SNPs that will actually be informative in your population sample. Again, it's gel-free, it's based on quantitative PCR, and it's even higher throughput. And then the highest throughput we can come up with that we're developing right now, and maybe next time I come back I can talk to you about some of that data, is actually pooled DNA SNP chip testing. So again, we're pooling DNA and actually running that on DNA chips and kind of using the data that the software puts out to actually extract quantitative data. Any of you that know about chip technology knows that they try and keep it qualitative. Basically, you either have a polymorphism or you don't. And the fact of the matter is the output from the program actually has quantitative data. You just have to know how to use it. So that would be, of course, ultimately useful. You can literally collect 100,000 genotypes, right, within about a week, which is pretty awesome. So it costs a little bit of money when you consider how much you have to spend in that 1 week, but it is orders of magnitude cheaper than doing 100,000 tests and paying for all those reagents and supplies. And again, any SNPs that are interesting, you could then expand and individually type in your population of interest. It's a very useful tool. So let me take a breath. We're going to switch gears now and talk about the analytic methodology. Okay? So when it comes to gene-gene interaction, there's really two methodologies that we'll explore. The first is kind of the less flexible, but it works very well for what we're looking at. Essentially, you can investigate two-way interactions, so looking at two genes or two polymorphisms together in one analysis. And essentially, you use cases only, and you construct a risk ratio. And really what you're doing is saying, does an individual that has two homozygous wild type or common variations, or two homozygous rare variations, does that product differ significantly from individuals that have one or the other? Now, if that deviates from unity or one, that implies that there's some kind of interaction effect between those polymorphisms. That works okay, and again, the limitation is that you can't look at primary effects, and you also can't model multiple interactions. So three-way interactions you can't, well, you can do, but it's not very powerful, as well as four-way interactions. So it's really good for two-way interactions, and the problem is that your multiple testing goes up, right? Even looking at 8 genes, you're going to do 7 comparisons, plus 6 comparisons, plus 5, plus 4, plus 3, plus 2, plus 1. So already you have to adjust for these multiple comparisons, and that becomes problematic, statistically speaking. There's a new method, set association, which is really promising. And the biggest benefit is the fact that it reduces what we call dimensionality, and I'll explain that in the next slide. And it's important in that it has the potential to capture a subset of all interactions that exist. Now, even though this is beneficial in that you can detect primary, two-way, three-way, and even four-way, in theory, if they existed, you could have higher interactions, but I'm not so sure that they actually exist. But unfortunately, there's no guarantee that you capture all the interactions. But again, I would tell you that that doesn't really matter. This is a much better method than not being able to capture even a subset. So the idea is you learn about which interactions you capture. You can then go back and ask more focused questions to identify new interactions. So the current methodologies can't handle large groups of SNPs. As I mentioned, too many variables are collected on individual patient samples. And it gets to the point where the number of observations in one sample outstrips the number of patients you have. And that's what statisticians refer to as the curse of dimensionality. Our statistics really aren't based on that. We really rely on the fact that the number of individuals outstrips the number of variables that you study. And really what that means is that testing each SNP for association yields a specific type 1 error for every test. And so you can imagine that if you look at their whole genome, even 100,000 genotypes, your type 1 error goes through the roof. You're not even sure when you get a significant result if it's real or not. There's not really a good way to distinguish. So the problem comes with the fact that complex traits are due to multiple interacting genes that are located throughout the genome. So what we should do is search for sets of marker loci at different genes and analyze them all together at once rather than individually. And then that would permit you to simultaneously assess both the primary, or what we call marginal genetic effects, as well as more global genetic effects on each trait. So set association is a simple strategy that allows for joint analysis of SNPs in different genomic regions. And now it combines information over SNPs by forming sums of information about each polymorphism. And it's actually information that you already know a lot about. By doing it globally, it reduces the overall type 1 error because the amount of comparisons you're doing is greatly reduced. And it limits the number of type 2 errors by combining this information together. So there's really 3 parameters that we look at. There's allelic association, there's Hardy-Weinberg disequilibrium, as well as using evidence for genotyping errors to exclude those SNPs which may be due to laboratory error, which can be really problematic. If you look at 100,000 genes, you can bet some of those are just purely lab error, and you want to be able to identify those. So Hardy-Weinberg disequilibrium is calculated in affected individuals and may be indicative of the presence of a susceptibility gene. So this is a chi-square test, right? Again, the straightforward statistical tool. Allelic association is due to the proximity of a SNP to a susceptibility marker. And really, again, it's a type of chi-square test, but really what it does is measures overrepresentation of genomic variants in cases versus controls. And while moderately high Hardy-Weinberg disequilibrium values in affected individuals may be indicative of genetic association to a susceptibility locus, very high values suggest some kind of genotyping error in the lab. So you can actually elect to create a cutoff. So if your p-value is, let's say, 4 orders of magnitude, 0.0001, you guess that that's probably due to some kind of lab error. And you take those out of the equation. You then send that back to your lab to be regenotyped. And in the meantime, you can analyze the rest of your data. So essentially, it's a lot of complex terminology for a pretty simple procedure. You take the statistic generated for allelic association, a chi-square 2x2 table. You take the statistic for Hardy-Weinberg disequilibrium, another type of chi-square, and you take the product, right? And that gives you a compound piece of information. And then you simply order them. So you say that value that's highest is number 1, the next highest product is number 2, and so on all the way down. Now, keep in mind that these two SNPs could be in the same gene, or they could be on completely different chromosomes. You're ignoring that relationship. You're just saying whichever one is the strongest gets to be first, and you go all the way down the line. And then the last piece of information is you start creating summary terms. So the sum of the first statistic alone, right, which is the exact same value. The next biggest statistic, you sum the 2 highest. And the next summary term, you take the 3 highest, and so on, until you've gone through all the SNPs. You then find the number of SNPs that are built into S, at which point you find a significant effect. So that boils down to a p-value. And really what you do to determine the significance level of that p-value is you use permutation. Now, for those of you that haven't really heard about it, permutation is a very useful tool now in statistics to kind of come up with appropriate p-values instead of using the tables in the back of our statistics books. And you simply randomize the test. You take your data, you break up who's a case and who's a control, and you kind of reshuffle the data and come up with p-values until you come up with a distribution. And I can talk to you about it more after the lecture if you're interested in that process. Now, set association relies on 2 assumptions. Okay? Again, every tool has a weakness, and it's important to understand them so that you don't make claims that are really beyond what your data shows you. The first is that the number of sums, S, so that means the number of SNPs that you identify, or polymorphism, that are involved in the disease are much smaller than the total number that you test. Okay? And it sounds very simple, but it's an important point. And the other is that interactions between SNPs will be captured as long as they flow into the summary term. So that means very subtle effects you might miss. And again, that may have to do more with looking at it again in a larger sample size. Okay? And again, these are weaknesses that are not uncommon among many different statistical tests. Okay? It's just important to understand your limitations. So those are really the tools that we plan to use to collect data in this population and do it in a way that's much quicker and, importantly, much cheaper than what we've been able to do in the past, which, again, is very important when it comes to human genetic studies or human studies in general. So the objective of the proposed research is to gain new insight into the idiopathogenic roles of prime candidate genes in HIV therapy, or rather their outcome. Now, the immediate, it's kind of listed in terms of what comes next for future projects. The immediate aim is to validate these findings in a larger cohort and to examine the entire sequence of candidate genes that we identify in this pilot. So if we identify polymorphism, say in the first candidate gene I listed on my list, we'll actually go back and resequence the entire gene to make sure that we identify all of what's going on to impact the risk of lipodystrophy. Okay? And then we'll apply the same methodology to the whole roster of candidate genes that we can afford to type that may modulate HIV severity and outcomes. And then, of course, future studies will propose to explore the impact of specific genetic variations in other measures, in quality of life measures as well as adherence. And that's actually what led me to the School of Nursing as a basic scientist, because nursing scientists are incredibly adept at integrating these kinds of measures, something that a basic scientist knows nothing about. And that's really where I see the advantage of that interaction. And we're to the acknowledgments. Clearly, this couldn't happen without an interest on both sides of the US. Here, the PIs of the P20 that funded this pilot, Professors Rivero and Holtzimer, my co-investigators, Professors Figueroa and Rodriguez Orengo, as well as our clinician colleagues, Doctors Merriman and Santana, for recruiting these patients. And last of all, my team back home. Hopefully they'll be able to see this webcast. I have a small family for my lab right now. It's really two people, Medical Carney with her husband and their new 6-month-old baby, Nikhil, and Leona, who just joined our group, and her husband, Jai. So they're the ones that really make all of this possible by helping me genotype. And when I say I want to try this new method, they're the ones that really have to say, okay, fine, we'll develop it for you. So they really are what keep me afloat. And with that, I'll take any questions. I know it's a lot of information, so anything is fine.
In drug therapy, how similar do the therapies have to be, or are you trying to actually get complete matches? And then how are you going to monitor if people are actually found that?
I'd say at this level, the level of this pilot study, it's not that easy to control for. I kind of, personally, that's one of the first questions I struggled with, and I realized that since these drugs are metabolized by the same set of genes, really the only difference in the therapy might be how quickly they present with these comorbidities. And since this design doesn't really look at longitudinal data about how that might happen, I could look simply at individuals that had lipodystrophy or not, and not really look at what regimens might have accelerated that. That's something I want to look at if we get funded to do a large-scale study and potentially follow them over time to see if I can look at that information. So right now we're not powered to do that.
But monitoring is also a problem, I guess, in many of these multiple therapies.
And that's what I'm relying on the clinicians for. I mean, that's something I can only, I don't know enough to know beyond taking their word for it that they might be doing that. Yeah. But now I'm going to have to check again now that you mention it. Thank you.
When you look at your 8 candidate genes,
Mm-hmm.
do those genes have are close in terms of interaction network with the proteins that they produce? Or because that can create problems in terms of one affecting the other directly?
Absolutely. It's a very good point. And I'd say there's a chance with the third and the fourth, PPAR gamma and SREBP1C. They're both transcription factors. Those are the only ones that I think, that I know there's evidence of being organized in a network. That's a very good point, and that's something I don't know that we'd detect in a sample of this size, but it's something we'd have to look at. Yeah, I'm not sure.
Exactly. I think that would introduce codependencies in the statistical
in the data. Well, and of course, right. And that's actually why I elected to use set association, because there you're already going into the analysis, you're already cognizant of the fact that you won't detect all the interactions, that you'll detect a subset, whether that's because by chance they weren't included or if there's actually a mechanistic basis. And I'm not sure at this level of an obvious way to do that, unless at the next level, when we have a larger population, actually not looking for networks that are at least known, and then trying not to, or trying to account for that information. I'm not really sure offhand how to do that at the SNP level. That may be possible at the expression level.
Right.
Yeah.
In your set association, is the permutation through bootstrapping or Jackknife?
It's bootstrapping.
How robust is that test?
Well, if you read the papers, which is really what I rely on, It's supposed to be fairly robust. There's a series of papers that have kind of demonstrated that using various SAGE data sets. So as far as I know, that's what I rely on, since I'm only an informal statistician.
Same here, so that's why I asked the question. Because some of the permutation tests, depending on how they're applied, can be more or less robust.
Okay.
And so,
Yeah, I actually collaborate with Jur Gott, and he's the gentleman who developed the methodology. And I kind of, whenever I get those questions shot at me, I email him, and so far, he seems pretty confident, although maybe he's a little biased.
So in the permutation analysis,
Mm-hmm.
basically once you have a collection of genotypes for a population, you can use that method to determine associations for just about any disease, right?
Absolutely.
Once you have that population genotype.
Right. And that's actually something, that's part of my master plan that I didn't want to talk about. If you look at this, that diagram that I showed you, it's my way of kind of leveraging the data produced in my lab. So we'll look within each particular disorder, we'll look using set association at interaction effects there. But then, of course, to gain power, if I find more global, if I use more global phenotypic measures, I can then lump a lot of these groups together and really look for interactions that might predispose to a whole series. And I think I would find an overlapping but some unique subset of determinants that I wouldn't see only within each disease. So yeah, I intend to apply this methodology both within and across as many of the diseases as possible. Yeah.
Do you already have controls for your genotyping experiments?
Yes.
That everybody will use the same?
Yeah, and I didn't have time to get into that. I actually have some pretty interesting control subjects, both specific to each one of these diseases, but also I've been very fortunate, again, because of this tight clinical research interface. We've, through my old mentor for my postdoc, he actually recruited what we call a super control group, and these are elderly individuals that were recruited from Senior Olympics. So these individuals are in their 70s to 80s, and they're running the 100-meter dash, and they're incredibly fit. And that's kind of a way of looking at individuals that have selected for optimal genotypes, as well as normal controls, which is kind of a moving target depending on what trait you look at. So we're lucky in that there's a genomic resource I have access to that has about 20,000 patients, and about 1,000 of them are controls, and they're controls for different parameters or different traits that we look at. So yeah, for all of these, there are control groups. And in the case of lipodystrophy, the control is actually the individuals that don't develop lipodystrophy. Again, not a perfect control, but it's one that we use for now. Yeah.
question?
Thank you very much.